Now we come to a very, very important biochemical process which is concerned with our talk on absorption of amino acids, right? What is that cycle? Or rather, okay, I revealed it already. It is a cycle. What is that? It's known as gamma glutamyl cycle or the Meister cycle. I hope that is how they pronounce it. I don't really know. I'm not sure about my pronunciation. But I think that is what the pronunciation should be. M-E-I-S-T-E-R. Meister. Meister. Okay, I'm confused, alright. What's in a pronunciation? Alright. So whatever this is, M-E-I-S-T-E-R cycle. Right? What the hell is this about? From one moment we were talking about absorption of amino acids, the next moment we come to a cycle. What is this going on? Let me explain. See, when the absorption of amino acids is happening, a process is also going on, or rather a process which is linked to the absorption of amino acids is Meister cycle. It is also known as gamma, this is not Y, okay, let me write it, let me spell it out for you. Gamma glutamyl cycle. Gamma glutamyl cycle. What is this all about? Once this cycle is explained, you will be able to understand the correlation between this cycle and how it is linked to the absorption of amino acids. So this jump that I made from absorption of amino acids to this is not random, alright? This is related. You'll understand that. What is happening here? Obviously this is a cycle. This is a cycle. We can understand that. What is it related with? It is related with the absorption of amino acids. That also I have told. What is the one thing that you must remember about Meister cycle? You must remember about Meister cycle. Like if I tell Meister cycle, this name should immediately pop out in your mind. What is that? It is none other than gluta. Ever heard of this? Of course you have. Glutathione. We have heard about this countless times before. And what is this thing? This is actually a biomolecule. It is a bio means found in us. Bio means living. Molecule because it's a molecule. So it's a living molecule. So does this live? Is this alive? No, actually it is found in biological organisms. So it is a biomolecule. What kind of a biomolecule? It is a, yes, the thing that we are talking about. Protein. Yes, it's a protein. More specifically, can I be more precise? Of course I can be. It is a actually tripeptide. Tripeptide means, obviously you guys know that, 3 amino acids make up this molecule. What is this? Glutathione. What are the 3 amino acids which make it up? The 3 amino acids are glutamate, or rather glutamic acid, plus cysteine, plus glycine. Beautiful. Glutamate, cysteine, glycine. So what is the chemical or rather biochemical name of glutathione? Glutathione is kind of a formal name which is given to it. But if we go by the constituents, what will its name be? Very, very crucial to remember. Gamma glutamyl cysteine glycine. Yes, so it is gamma glutamyl cysteine. Okay, I missed an I here, I'm sorry about that. Yes, it is EI. Yes, because obviously this is EI here, so obviously it is EI here also. Gamma glutamyl cysteine glycine. If I say Meister cycle, okay, whatever it is, this cycle, this is the name that immediately pops out in your mind. Why? Because this is the pivotal molecule in this cycle. How is that? What is happening? Very well, let's take a delve into the real cycle now. Alright, this is actually linked with the absorption of amino acids. This is perhaps the 1000th time I'm telling that line, because I cannot think of anything else to tell right now apart from the real cycle itself. So let's talk about the real cycle itself. Alright, now the real cycle, what are we talking about here? Meister cycle. This is going on in where is this going on in? This is going on inside the cells. So perhaps I'll be able to explain it to you here. A line. Alright, what is this line? This is the cell membrane. Oh, it's going out of the camera, I'm sorry. This is the cell membrane. Does it sound new? Of course it not. So it's the cell membrane, right? Where is the amino acid right now? Outside the cell. This is inside the cell, that is outside the cell. We have just finished digestion. Now we need to transport this amino acid from outside the cell to inside the cell, right? So to do that, that is where this molecule glutathione comes into play. Alright, that is where the molecule glutathione comes into play, and that is where this entire cycle, that is the Meister cycle, is generated. Alright, so what happens is glutathione where is it? Outside the cell or inside the cell? Yes, inside the cell. Very good. So inside the cell is glutathione. Outside the cell is amino acid. Their reaction needs to take place. But how can it take place if both are in different regions? Amino acid is outside the cell, glutathione is inside the cell. They need to be at a common place for the reaction to take place. What is that common place? Yes, that common place is inside the cell, not outside the cell. So glutathione does not move outside the cell and react with amino acid. Amino acid is transported inside the cell and it reacts with our friend glutathione. So the enzyme which is required in this reaction is gamma. This is not Y, alright? Gamma. Just telling. Gamma glutamyl. Okay, it's becoming congested. Gamma glutamyl transferase. Gamma glutamyl transferase is the enzyme which is actually being used here. That enzyme is leading to, or rather it is catalyzing the reaction between the said amino acid and glutathione, right? Now, what is formed in this reaction? Listen to me very carefully. This reaction forms glutamyl amino acid. Alright, now give me a patient hearing. What was glutathione made of? We just said a few minutes ago that the constituents of glutathione are what exactly? Yes, gamma glutamyl cystenyl glycine. Gamma glutamyl cystenyl glycine. Here, the compound which is formed, the molecule which is formed, is glutamyl amino acid. Don't you think something is missing here? Yes, of course. Where did the cystenyl glycine part go? Where did the cystenyl glycine part go? Answer is, it is there. It's just been separated, that's all. So the cystenyl glycine remains. Cystenyl glycine has been separated from the glutamyl, alright, the glutamate, and that has actually combined with the amino acid, the said amino acid, and glutamyl amino acid has been formed. Alright, now we go on to the next step. Remember, this is a cycle, so we will come back to glutathione, so that is why this slanted arrow. Now this said, in the next step, what happens is another enzyme comes. Again a gamma, it's not a Y. There is no Y here, alright? Gamma glutamyl cyclotransferase. Right? Gamma glutamyl cyclotransferase. It's pretty hard names. I can understand that, but what can we do? Have to remember them. At least for the time being, we have to remember them. Interesting step. This is an interesting step. Glutamyl amino acid was formed, right? And at the very next step, the amino acid is released. So the amino acid was here, which was outside the cell, and glutathione reacted with it, formed glutamyl amino acid, and released the amino acid. So now the amino acid is, yes, mission successful. Amino acid is inside the cell. It was outside the cell. Now it is inside the cell, which was our objective. So our objective is fulfilled. Yes, great job. But unfortunately, the cycle must go on and the glutathione must be regenerated, because obviously that is why this is a cycle at all, right? So that is exactly what is going to happen. Our objective is completed, but we need to finish this cycle so that glutathione can be regenerated and it can react with the next amino acid and transfer this amino acid, the next amino acid, from outside the cell to inside the cell, right? Right. So now glutamyl amino acid, gamma glutamyl cyclotransferase gives a beautiful compound, oxoproline. Oxoproline, quite the name, right? Oxoproline. Now oxoproline is formed, but I like to think of oxoproline as an intermediate. Why I do that? See, I told you that the cystenyl glycine part has been removed, and what remained was the glutamate part. The glutamate part remained bounded to the amino acid. Now the amino acid has been released, so what should remain is the glutamate. But no, what remains is oxoproline. So let's think of oxoproline as an intermediate, right? The next thing that oxoproline gives is glutamate. Now this makes sense, because I just now said that let's consider oxoproline as an intermediate. So if oxoproline is intermediate, it leads to glutamate, right? What is the enzyme that is needed here? The enzyme is 5-oxo 5-oxoprolinase. Oh my god, this is very bad. Congested. 5-oxoprolinase. Now, can it be understood? I hope it can be. 5-oxoprolinase, right? Right. Now, this glutamate is formed. Now the things that will happen is very beautiful. See, you guys will be able to tell this to me yourselves. We need glutathione. We have glutamate. What else do we need? We need this. We need this. The cysteine glycine is the thing that we need. So cysteine glycine will react with glutamate and it will form glutathione. Right? Well, right, but then again, cysteine glycine does not remain like that. It will break down into what? Glycine and yes, cysteine. This cysteine will react with glutamate. First, it will lead to the formation of glutamyl cysteine. Easy enough, right? Cysteine reacts with glutamate. Cysteine reacts with glutamate and it forms glutamyl cysteine, right? Then what happens? Very easy. Glutamyl cysteine. So the next thing that happens is the glycine also reacts with the glutamyl cysteine. And what do we find? Yes, glutathione. So that completes the cycle. Oh wait, we have some incomplete things to take care of. The enzyme names. Yes, I left these enzyme names for a special purpose. You see, the first enzyme that we encountered was gamma glutamyl transferase, right? And that makes sense. Why? Because transferring the amino acid from outside the cell to inside the cell is what this enzyme is doing. So it is gamma glutamyl transferase. It makes sense. The compound which is being formed, the molecule which is being formed is glutamyl amino acid. Glutamyl amino acid leading to the formation of cysteine glycine also. Means cysteine glycine actually gets broken down from the glutamate. The glutamate and the amino acid remain bounded to each other. Next, the amino acid is given out and we find an intermediate which is oxoprolin, which by the enzyme 5-oxoprolinase, and by the way the previous enzyme is gamma glutamyl cyclotransferase, we find glutamate. Now, what is this? Glutamyl cysteine. Yes, the enzyme name here is glutamyl cysteine. Yes, synthetase. Yes, glutamyl cysteine synthetase, right? And following the same logic, what is the name of the enzyme here? Yes, I'm giving you 5 seconds. 5, 4, 3, 2, 1. Time over. Yes, absolutely correct, or maybe not correct, whatever your answer was. If your answer was glutathione synthetase, congratulations. And if not, well, better luck next time. Anyways, so this makes sense, right? This actually makes sense. Synthetase. This is one, odd one out. 5-oxoprolinase. Do me a favor and please remember the name of this enzyme. And the previous two are transferase. Gamma glutamyl transferase, and this is gamma glutamyl cyclotransferase. Now, just do me one last favor. Alright, one last favor. No more favors after this. one last favor and remember this one thing, that this cycle requires 3 ATP molecules. So remember what I said, that the absorption of amino acids is ATP dependent. I had said this. Of course I had said this. Come on, you can't deny that. So I had said this. Now we are proving why this is ATP dependent. Because from transporting this amino acid from outside to inside, 3 ATP molecules, 3 ATP is required. Where are those 3 ATP? Have I given them in the cycle? No. So that means they are not there? Of course not. They are there. I forgot to give them. Come on. So where are those 3 ATP being actually used? From ADP to ATP, where is that happening? At the last 3 steps. The last 3 steps. ADP. Oh, I'm sorry, not ADP. ATP to ADP. Again, ATP to ADP. Again, ATP to ADP. The last 3. We'll be explaining this later. This heart nerves disease, we will be explaining this later when we have dealt with most of the metabolism. Alright, this is one. Then again, there is another thing called oxoprolinuria. Oxoprolinuria. What is that? Deficiency of this enzyme. This is easy, right? This is easy. Deficiency of this enzyme, that is 5-oxoprolinase, leads to a condition known as oxoprolinuria. Right? Right. Now, there are kind of many other clinical applications of this also, but as I have said, they will be dealt with later after we have understood the specifics of protein metabolism and we have gone through most of the amino acids individually, their metabolism. So that will only make sense after that.